Hello everybody. Okay, we're back. Hopefully this will be the last part. We can knock out this last chunk. This is the newer material. So this will be potentially what will be a little bit more of the focus on the final. I know it is technically cumulative, but more than likely you'll see a decent chunk of the newer material on that. I believe this is the newer material at least. So gluconeogenesis. So when we talk about gluconeogenesis, specifically we're talking about creating glucose or creating energy from the non-glucose sources. So stuff like our proteins and our fats. Because as we talked about before, glucose really is the energy source for the entire body. Parts of our body like the nervous system, the brain, and even the muscles as well, like they need glucose in order to survive. And so if we don't have any available to us, or let's say even if it's just like we haven't eaten for 6, 7, 8 hours, and we have other energy stores like fat or proteins, our body is going to use them to maintain our blood sugar, maintain our levels, make sure that we are that our body and the very specific parts of our body have enough energy to do what they need to do. Okay? So, like we said, this is glucose synthesis from non-carbohydrates. It's in order to meet the needs of the body for glucose during carbohydrate insufficiency. We need a continual glucose supply as it is the energy source for the nervous system and red blood cells, specifically these two. That's the important thing to know. These can only use glucose. Glucose only. Each molecule of glucose produced from pyruvate requires the 4 ATP molecules, 2 GTP, and 2 NADH molecules. So this is talking about when we're going backwards, like producing glucose from pyruvate. This is what we need. The allosteric regulator, we have fructose 2,6-bisphosphate that prevents wasteful operation of both pathways at the same time. So we can see splitting between gluconeogenesis and glycolysis, continuing through glycolysis, or going back to gluconeogenesis. So the conditions that activate the process of gluconeogenesis, we have the fasting or the starvation state, which is about not eating for about 8 hours. A low carbohydrate diet, meaning that glucose itself is not as plentiful as it normally is, as well as intense exercise, if we've been exercising either really, really hard or for a longer period of time, we can use up our glycogen stores, and then we're pulling the glucose from the blood, and we need to get more into the blood in order to meet those energy demands. So the importance of glucose is that glucose is the only fuel for muscles in anaerobic conditions. It's a precursor of milk sugar, lactose in mammary gland. It is used to clear products of the metabolism of other tissues from the blood, so like the lactate, glycerol, propionate. All of these things will be used to be converted into glucose, whereas otherwise they're just waste products floating around in our system. Okay, so some of the substrate materials for gluconeogenesis. So any compound that can be converted to either pyruvate or oxaloacetate can be a precursor for gluconeogenesis. So if you think back and remember when we were looking at that diagram, you kind of had like the main wheel, and we had like oxaloacetate, malate, all those different areas when we're talking about what the different intermediates of the Krebs cycle could become. And this is kind of talking about it now going the opposite way of these certain things can then turn into the intermediates that we need to continue to produce energy. So like alanine can be converted to pyruvate, aspartate can be converted to oxaloacetate, and glutamate can be converted to alpha-ketoglutarate. And we also have all of these as well. These are all potential sources for gluconeogenesis. All of these can be converted back into glucose. So gluconeogenesis, where does it take place? Primarily in the liver, and then a little bit in the kidneys, as well as technically some of the epithelial cells of the small intestine. More than likely, the question you're going to get is asking something along the lines of, does gluconeogenesis occur in the muscles? And the answer to that is no. Or it will be, where does it primarily take place? And that would be the liver. So the pathway of gluconeogenesis, the enzymes that bypass the reverse pathway of glycolysis. So when we're talking about glycolysis, there were those 3 specific enzymes, those rate-limiting steps, that those are the ones that are irreversible and they can't go backwards. So we have to find a way to kind of loophole around those. And that's where these specific enzymes come into place. So in place of hexokinase, glucose 6-phosphatase is used in order to bypass that step. For phosphofructokinase 1, we have the fructose 1,6-bisphosphatase 1. And for pyruvate kinase, we actually need 2 separate enzymes, pyruvate carboxylase and PEP carboxykinase, in order to get past those specific steps. And just like how these are the steps that use ATP to go through them, these other ones are going to be, specifically down here, these ones are going to be using all that ATP to go backwards. And these are the exergonic and irreversible reactions. These are the typically irreversible reactions. So again, they have to go backwards. So you'll want to know those 3 specific reactions. And I would also recommend knowing which enzyme they help bypass. Okay, pretty short chapter there. Again, that's all that's really talking about in gluconeogenesis. If you have any more questions about gluconeogenesis, or you want to review that, you can go back and review the other YouTube video that dives deeper into all of the reading and all of the book that we had. Okay, lecture 15, ethanol metabolism. So ethanol, specifically we're talking about ethanol is synonymous with alcohol. We're talking about the metabolism of alcohols. So alcohol dehydrogenase, the function of the reaction, and the oxidizing agent use. So first and foremost, it gets the enzyme alcohol dehydrogenase catalyzes the oxidation of ethanol to an acetaldehyde. And it's using NAD+ as the oxidizing agent. So this oxidizing agent, remember ethanol gets oxidized, so that the oxidizing agent is the NAD+. So it's going to, NAD+ is going to be reduced. The oxidizing agent gets reduced to NADH. So it takes off that H to make it acetaldehyde. And then we have our acetaldehyde dehydrogenase, which then oxidizes acetaldehyde to form acetate. Same exact step, where NAD+ is the oxidizing agent. Very, very similar. As we go from acetaldehyde now to acetate. And we've talked a little bit about this, about kind of our primary, secondary alcohols a while before. And so this is really kind of digging into those specific functions, the specific actions that we had talked about with those for like primary, secondary alcohols being oxidized. So when we talk about chronic alcohol consumption actually increases the ratio of NADH to NAD+. So actually this would be in our brackets because we're looking at the concentration. And that's actually why alcohol, especially chronic alcohol consumption, can be so dangerous. So we can look at the effects, because we have this high levels of NADH. So then let's look at kind of what that causes. We can have lactoacidosis, which is an increased production of lactate in the body. So the pyruvate is being turned into lactate. And we also talk about with lactate, how does lactate cause, or what does lactate cause to happen in the blood? We talk about lactate is also a lactic acid, and as an acid, we have a whole bunch more hydrogen ions, and that will actually cause the pH to drop. So we also have inhibition of gluconeogenesis, with the low levels of pyruvate inhibit the gluconeogenesis. Depletion of liver glycogen in more severe cases, which can lead to a resultant hypoglycemia. And you also get a decrease in the citric acid cycle activities. Because the pyruvate is being converted so quickly to lactate, we don't have because it's converted to lactate because we have so much NADH around. So you don't have pyruvate. Because you don't have pyruvate, you don't get acetyl-CoA. And with no acetyl-CoA, you don't get the process, you don't get it going into the citric acid cycle. So kind of to combat alcohol, we have this function, we have this microsomal cytochrome P450 system, also known as the microsomal ethanol oxidizing system, MEOS. Okay, you'll see these referred to as P450 or MEOS. And that's going to generate acetaldehyde involving the NADPH. So these MEOS increases in chronic alcoholism, and it increases the blood levels of acetaldehyde and acetate. Let me actually find, the book does a really good job. There's a short chunk on this, because I don't feel like what I have written here does quite a good enough job going over this. This is the ethanol. The book is almost identical to that, so let's actually do this. The P450 because we don't want to get too deep into it, but we'll look at this. So more generally, this cytochrome P450, or this whole system, is involved in the metabolism of drugs, chemicals, and endogenous substrates. So really, I guess the big picture that you're going to want to know from this is that the P450 system is responsible for helping to break down the alcohol and helping to detoxify the blood and everything from it. Okay, and so if we have a whole bunch of ethanol, this P450 system is going to be, these specific enzymes are the ones that are going to be breaking down the ethanol, breaking it down and oxidizing it from ethanol to acetaldehyde, and then from acetaldehyde to acetate. That's its job. And that's really all that there is in that chapter. Pretty quick. So now getting into the structure of nucleic acids and DNA replication. So DNAs and RNA store genetic information. Genetic information is the code for all of our, we'll call them phenotypes, which is how our body presents, like what it actually presents, what proteins it makes, which like ends up determining all sorts of how ends up determining how our body works, how our metabolism is, even the color of our eyes, color of our hair, etc. So some definitions: we have replication, where DNA is making DNA; transcription, where DNA is used to make RNA; and then translation, which messenger RNA is translated to form peptides, peptide chains, polypeptide chains, which turn into proteins. So the chemical structure of DNA and RNA and some of their differences. So both of them are made up of nitrogenous bases. Adenine, guanine are our purines, and our pyrimidines are cytosine and thymine, and also uracil. Uracil takes the place of thymine for RNA. We also will have a sugar, a deoxyribose or just a ribose, and a phosphate. You can see this is our full structure of our nucleotide. The phosphates are cool because they allow actually the different bases. They allow the bases to bind together. We'll have peptide bonds between the different nitrogenous bases of the DNA or RNA structure. So the difference. So this is some vocabulary again. Nucleoside versus nucleotide versus nucleic acid. So a nucleoside is just the base plus the sugar. Nucleotide is the base plus the sugar plus the phosphate. And then the full nucleic acid is the RNA or the DNA. So you can see kind of the difference. The ines, adenosine, guanosine, cytidine, thymidine, and uridine, these are our nucleosides. So IDE ends with INE. Our nucleotide ends in the 8, and then our nucleic acids end in the DNA or RNA. So the linkage, we're talking about actually building them now. Linkage is joining the nucleotides within a nucleic acid strand. You have what's called a phosphodiester linkage. And it's going to join the 3 carbon of one sugar to the 5 carbon of the next sugar. And we can see that where the 3 carbon on the bottom versus the 5 one on the top. So it's just the easiest place for it to bind. A phosphodiester bond, essentially what it means is phospho means that there's phosphorus, and the diester means the ester bonds are typically with an alcohol with an OH group. And you can see right here how there's an H coming off this carbon, H coming off of this carbon. So that's the ester linkage. Know what the type of bond links two different nucleic acid strands or the complementary strands. Those are hydrogen bonds, so between an A and a T or an A and a U for RNA, there are 2 hydrogen bonds for each of those. Between the G and the C, there are 3 hydrogen bonds. And this is known, comparing A to T and G to C is known as Chargaff's rule. That the total number of adenosine and of A and T are going to be equal because A will only bind to T. Same thing with no, not necessarily, never mind. I was going to say the same thing as RNA, but it's not quite because RNA tends to be single-stranded. So that's different. But with DNA, specifically, let's see, actually change this. Chargaff's rule is specific to DNA. So within a double-stranded portion of DNA, the total number of A's equals the total number of T's, and the total numbers of G's equal the total number of C's, because they're always going to be bound together. Looking at the structure of DNA, we have these different structures: bDNA, aDNA, and zDNA, and also nucleosomes. Most common that we're going to talk about is bDNA. This is like our standard DNA that's loose. It's a right-handed double helix. Nucleosomes, we'll talk a little bit about when we talked about this in the last couple of chapters, with kind of how DNA is stored and wrapped up. Because if you think about how much DNA that there actually is in our bodies, there's so much, and it's it would be really, really long. So in order to store it properly, you have these histones, these proteins, that help to hold and wrap the DNA. So a nucleosome is what we call the DNA wrapped around that histone core. That's when 8 histone proteins all come together into a disc shape to wrap and essentially roll up the DNA. So histones can be acted on to perform different functions, such as acetylation. If the histones get acetylated, it's going to unwind the DNA and allow for gene expression. Whereas methylation, if they get methylated, that's going to lock them down, and it's going to suppress those genes. Denaturation and renaturation of DNA. We kind of have already talked about this as well. Denaturation is the breaking down of proteins. Renaturation is them coming back together. And it depends on stuff like pH and heat. And we talked about this earlier in a previous chapter during this review as well. So talking about DNA replication. So DNA replication is bidirectional. The DNA synthesis always occurs in the 5' to 3' direction. Make sure you read the questions really, really careful when you get asked them, because the strand that is being formed gets built in the 5' to 3' direction. But if the question is asking how is it, what is the direction the strand is read when building a new strand, it's reading the other strand 3' to 5'. Hopefully that makes sense. He can ask the question, or the professor can ask the question either way. So it's good to kind of know it both ways. Replication starts at a site of origin and moves out in both directions from this point. DNA replication is semi-conservative, which means that the following replication, each daughter DNA or new DNA pair have one intact parent strand and one newly synthesized strand joined by base pairs to the parental DNA. The reason also why it's semi-conservative is also to kind of protect the DNA, because if you think about if it had to reform both new strands and then split them and then put them together, that just leaves more room open for error and for potential problems to arise. So we have the Okazaki fragments. The Okazaki fragments are these fragments here on the lagging strand. The leading strand, it gets built straight across. The lagging strand is going to be built still 5' to 3' direction, but because it's having to read it opposite, it forms a little chunk, and another one forms a little chunk, and then they get together, and then these will later be spliced together by DNA ligase. Constructing a complementary DNA strand. So this is our little quick step-by-step. So DNA helicase unwinds the strands. Topoisomerase prevents extreme coiling. RNA polymerase or primase, depending on either one can do this job, is going to lay down an RNA primer. This is going to allow for the DNA polymerase to bind. to where it needs to go. It's going to bind, and it's going to form a new strand on the leading and the lagging strands. So it builds the new DNA from 5' to 3'. It reads the old DNA, 3' to 5'. And the Okazaki fragments are built in the lagging strand. DNA ligase joins the Okazaki fragments together on the lagging strand. So the big difference between RNA is that it's going to use RNA polymerase instead of DNA polymerase to build its structure. Knowing the function, so here's all of our different enzymes that we use. We have DNA polymerase, RNA polymerase, DNA ligase, the topoisomerase, helicase, and also exonuclease and endonuclease. Our exonuclease and our endonuclease are responsible for combating the possible mutations. So if we have some kind of mutation of the DNA strand, let's say we have a spot in the very middle. Let's say we have a spot in the middle that we had a mutation, it's like something got put in a little bit wrong. Our endonuclease is going to come and kind of snip that out. So it's going to snip the mutation out and then allow for the DNA polymerase to come back and put in the right one. The exonuclease is kind of like the backspace button, where it's going to start at the end of the strand and break it back down, going backwards until it reaches the messed up part, and then it will put in the DNA polymerase will put in the correct one and continue rebuilding the DNA. Lecture 17, talking about RNA structure, specifically transcription. We talked about replication, now transcription. So the chemical structure of RNA is very similar to DNA, just has the ribossugar, the phosphate, the purine, the pyrimidine base. Remember, this one has the uracil instead of the spacing on the word, in place of the thymine. The linkage that joins RNA, we have the phosphodiester linkage in the RNA, the 5' to the 5' to 3', just like in DNA. The base pairing, everything's the same except we replace the T for a U. Function of the different enzymes, we have ribonuclease, which cleaves the RNA molecules and catalyzes the breakdown of RNA into smaller components. Remember, eventually after we finish using an RNA, we're going to want to break it back down. So the ribonuclease will help break it down later on. Peptidyltransferase, it's an enzyme in protein synthesis that consists of RNA, and it catalyzes the two principal chemical reactions of protein synthesis, the peptide bond formation and the peptide release. And ribozymes, these are the precursors of ribosomal RNA or rRNA. And they catalytically removes the internal segments of themselves, splicing the ends together. So it's going to, the ribozymes are going to have their act on themselves in order to pull themselves together in order to form the rRNA. Again, we have rRNA, mRNA, and tRNA, each doing their specific job. If you do not recall a lot of like what these guys do, go back and watch the video specifically on this lecture. I go through each of the types of mRNA, the full transcription, everything like that. We have the different RNA polymerases, RNA polymerase 1, 2, and 3, where they're located and kind of what their function are. So polymerase 1 is the precursor of most rRNAs. The polymerase 2, this synthesizes the mRNA. So the precursors of most rRNAs are the ribozymes. RNA polymerase 3 synthesizes a variety of the small nuclear and cytosolic RNAs and tRNAs. RNA synthesis occurs 5' to 3', just like DNA. Then we have these steps in RNA synthesis. So we have initiation, where it's initiated at specific DNA sequences called the promoter region. These promoter regions can be the CAAT box, which signals the binding site for the RNA transcription factor, and the TATA box, which indicates where a genetic sequence can be read and decoded. It's going to define the direction of transcription as well. So both of these, so the CAT box is the binding site, the TATA box is where the genetic sequence specifically can be read, where there's actually an active gene. Elongation. First, you have to unwind the DNA. Again, we use the helicase for that and the topoisomerase. And then the RNA polymerase builds an RNA strand. It's going to bind, and rather than just building a primer for DNA polymerase to come in, it's going to attach on and just go and build the RNA. The termination, this is signaled by a specific DNA sequence. Typically, it's a long A, like that, a whole bunch of A's in one spot. And the complementary sequence is going to be a bunch of uracils. And this causing of like this continuous U actually will help to release the RNA off. There's also something called a Rho factor. Rho factor is also going to be released and help to terminate this DNA signal, or the RNA polymerase, and help detach the RNA as it gets closer to the end of the gene that is trying to express. Now the post-transcriptional processing, so once we have a strand of RNA, there's only like specific bits or specific chunks of the RNA that actually code for the gene that we want. So the exons are those regions of the RNA that actually code for the gene or for the protein that we want. The introns are kind of like the extra fluff. You can think of them as a buffer within the DNA to help prevent the DNA, or so that it's less likely that a mutation will occur in the coding region of the RNA. Okay, so the DNA has these kind of non-coding specific regions, and so the RNA, because it has those specific regions, it helps prevent the DNA itself from, even if there is a mutation in the DNA, it's less likely to really affect the genes. But we just need to splice those parts out. So the exons are cut out and then joined together before translation. And that's for the formation of our mRNA. And now finally, talking about translation, actual building of the proteins. So genetic code, what is it? It is a collection of codons that specify all the amino acids found in proteins. It's like all of these guys. A codon is 3 specific base pairs. And each codon is going to pair with something called an anticodon on the tRNA. The tRNA is going to be the carrier for the amino acids. It's going to code and find the correct anticodon to go with the codon. And each of those specific anticodons are built to carry a very specific amino acid. So the codons are a sequence of 3 bases in mRNA. They code for a specific amino acid, and each of the 20 amino acids have at least 1 codon. A couple have multiple. Most important to know is methionine. Methionine is always going to be the first. It's called the start codon. It's always going to be the first one. The anticodons are the sequence of 3 bases on tRNA, and they're going to bind to the codon and bring the amino acid along. AUGs are start codon, which codes from methionine, and our stop codons, UGA, UAG, and UAA. The RNA that contains the anticodons is known as aminoacyl tRNA. Aminoacyl, this is literally just amino is like a kind of a placeholder for whichever amino acid specifically it's carrying. So like if it's an AUG, it pairs with AUG, it's going to be carrying methionine, and so this will be a methionine tRNA. It codes for whichever or it's named after whichever amino acid it's carrying. The RNA contains a codon is the messenger RNA. The function of the amino acid tRNA, it brings the amino acid and the anticodon to form polypeptide chain. We need to know the function of the amino acid tRNA synthetase. Amino acids are activated and attached to their respective tRNAs by amino acid transfer reaction catalyzed by the amino acid synthetases. So the amino acid synthetases are what helps to attach the amino acids to their paired tRNA in order for them to take it then to pair onto the codons of the mRNA and leave behind the amino acid to build the chain. The steps of translation. Let's actually look at here. We can see kind of what happens here. And if you go to the book, the recording that in the book, I go into a lot greater detail specifically about what happens here. But essentially, these here are our different subunits for our ribosomal subunits. And we're going to have a large and a small subunit. The larger, essentially the larger and the smaller subunit are going to come and they're going to sandwich the mRNA at the start codon, the AUG, and it's going to be in what's called the P site. And then our first, make sure that I have, okay. So the first ribosome part, there's going to be two different ribosomal parts. The first one attaches, and then our tRNA carrying methionine will come and attach in the P site. Then our larger part of our ribosome will come and sandwich in between. Then it's going to get a signal where the next, if I can find, this picture is a little bit confusing. It's easier in the book. Let me pull it up here. It's been a while since I made that specific flow chart, so I want to make sure that I here we go. It's a little bit better. Okay, so our 40S subunit, that was also what was wrong. We have a 40S and it's supposed to be 40 and 60. Yeah, it's supposed to be 40 and 60. For some reason it has it 30 and 50, but it's 40 and 60. So this should be a 6. Okay, so the 40S subunit will come and bind to the smaller ribosomal subunit will come and bind right on this initiation codon, the AUG. The first tRNA that carries methionine comes and attaches, and then our 60S subunit is going to come and sandwich between the two of them. I'm fully setting it up. So there's an E site, an exit site, a P site, and an A site. A site is where it enters, E site is where it exits. So next, our income, we'll have the amino acid tRNA, whichever one it specifically codes for, is going to come in and enter onto the A site and link in the anticodon pairs with the codon. Then the amino acids that they're carrying are going to bond together, and then they're going to shift all one to the left, or well, the ribosome will shift down, so then what is in the A site moves to the P site, and what's in the P site goes to the exit and will come off, and then our new one comes in. And that's how it's going to be. It's going to continue just like that. Finally, at the very end, when we get to one of our stop codons, like UAG, we have a release factor, and that's specifically going to be coded for it. And the release factor comes and binds and effectively ends the process and releases the polypeptide chain. And then that polypeptide chain can go off and fold and do all the stuff that it's supposed to do. I go through every single one of these steps in the other video. So if you're really confused about this and you haven't watched the other video, go back and watch that other one where I go really deep into detail about each of these things. You'll want to know this process fairly well. Maybe not necessarily like the intrinsic factors and stuff, but specifically the step-by-step of the subunits, kind of which subunit, so the 40S subunit is binding first, the 60S subunit binds second. What is an anticodon? What is a codon? Where are each of those? Kind of how this, in general, how this process of elongation occurs. So these are forming, the amino acids form peptide bonds. And then the inhibitors and their mechanism of action to distinguish them. So we have puromycin, which inhibits the peptide bond formation. We're going to do block translation by labeling and releasing elongating peptide chains from translating ribosomes. And chloramphenicol, which inhibits the peptidyl transferase, and it binds to the 58 to the 60S ribosomal subunit. So just know, I would know the names that these specifically inhibit the elongation and peptide bond formation.